first I want us to get on the same page with the etiologies of Parkinson's disease. So if you have Parkinson's disease, well, either you have a sporadic form of Parkinson's disease for most of the patients, because both genetics and environmental factors, so multiple factors, contribute to the disease. On the other hand, for around 10% of the patients, they're going to have what we call a monogenic form of the disease, meaning that if they have a mutation in one gene, they're going to get the disease. And this is mostly what we're going to be interested in today. So I just displayed here 4 genes, SNCA, LR2, PINK1, and Parkin that I'm going to talk about later, but we know about 15 genes so far. But at this moment, you might wonder, how do I know in which box I am if I have Parkinson's disease? Well, we know that the earlier the onset is, and the more family members are affected in one family, the more likely we are to be on the 10% side. But what we know is that if we do a targeted gene sequencing of those 15 genes, we only identify a diagnostic in 10%, meaning that likely there are more genes to discover in Parkinson's disease. For years and decades, the Paris Brain Institute collected samples from individuals with Parkinson's disease and their relatives. Those patients underwent multiple genetic testing. For some of them, we identified, of course, diagnostics, but for a lot of them, we were missing a diagnostic. And we know in the same time that on the other side of the Atlantic, at the Center for Alzheimer's Disease and Related Dementia, at NIH, they have a good experience with long-read sequencing. And at the Paris Brain Institute, we have the cohort. So we were like, okay, let's do a collaborative project. And we shipped samples from the Paris Brain Institute to the NIH. I went there, sequenced the sample, and in total, we did long-read sequencing on 106 individuals with early onset and huge families of Parkinson's disease. I'm not going to go too much into the details of the protocol and the QC in the interest of time, but you can scan the protocols we've published in August if you want to know more about it. Our aims, were to reach 25 kb to 30 kb N50 and to get a throughput of 100 to 120 to get a 30x at the end. Overall, we were satisfied with our wet lab protocol, and what we get after loading the flow cells in the P24 and the P48. So we were able to move forward with the bioinformatic pipeline. Again, I don't have time to go too much into the details of that, so that I can have time to show you the 2 examples showing how long-read sequencing can be useful for individuals. This first case is mostly going to be related with clinical aspects, and the second one, research aspects. So we had this family with 2 siblings affected by the disease. We were thinking of an autosomal recessive inheritance. They had a quite atypical Parkinson's disease. They had an early onset, 31, 33 years old, a good response to levodopa. One of them had also a dystonia. They had a slow progression, as we can see, with a low UPDRS score after 15 years of the disease onset. So for a trained neurologist, all that is suggestive of a PD related to Parkin. Parkin is the gene mostly involved in early onset and familial Parkinson's disease, and it's an autosomal recessive gene, meaning that you need 2 mutations to get the disease on both alleles. And this is important for what's coming next. So with this family, we first, with the Parkin in the back of our mind, performed Sanger sequencing and multiple ligation probe amplification, MLPA for short, and we identified 1 copy of exon 4 of Parkin, which was interpreted at that time as a heterozygous deletion of exon 4. We further performed multiple exams, another MLPA, a targeted gene panel sequencing, whole-exome sequencing, ddPCR, but we only had 1 heterozygous deletion of exon 4. At least this is how we interpreted it. So we had no diagnosis until 2023, where we performed ONT long-read sequencing. And what we identified is quite interesting. So we have here a deletion, heterozygous deletion, encompassing both exon 3 and 4 on one allele, and on the other allele, here in blue, we have a duplication of exon 3. So overall, as the other genetic testing said, we have 1 copy of exon 4, and we have 2 copies of exon 3, which is normal. Here, we all have 2 copies of exon 3, I mean, I hope. But so with this, it explains why the other genetic testing weren't able to identify this. And as I highlighted with the broken lines in red and blue, the breakpoints were deep intronic, and whole-exome sequencing, of course, couldn't identify it. So first case, long-read sequencing is able to unravel the complexity of rearrangement in already known PD genes such as Parkinson. So the second case is going to highlight how, with long-read sequencing, we can discover new genes of Parkinson's disease, because, as I said, in the beginning, only 10% of patients with early onset and familial Parkinson's disease get a diagnostic. So we had at the Paris Brain Institute this huge family with 7 members affected. Again, we performed multiple exams, no diagnostic. So what we did, ONT long-read sequencing. And we identified a 65 kb deletion, heterozygous, of a gene named PD10A. So I'm just going to talk a little briefly about PD10A, and then I'm going to go back to the family again to show you what the segregation analysis revealed. So we had this deletion encompassing exon 3 out of 22. It's a frameshift deletion that is leading to a stop codon at exon 4 out of 22 of the protein, of the gene, sorry. And as you can see in red, the 2 breakpoints were deep intronic. This is why, again, whole-exome sequencing couldn't identify this deletion. This deletion is absent from control database, and PD10A is a highly constrained gene, meaning that it doesn't like to get loss of function mutations. And more interestingly, as you can see here, this gene is expressed mostly in the brain and in the caudate nucleus and the putamen, which together form, in pink here, the striatum, which is the region which is receiving dopaminergic neurons projection from the substantia nigra. And in Parkinson's disease, the neurons that degenerate are the dopaminergic neurons. I'm not going to go too much into details of the biology of PD10A, but it's an enzyme that is degrading both CAMP and SYNGMP in the medium spiny neurons of the striatum. What do we know in the about the clinic of PD10A at the moment? This gene was reported in 31 individuals so far. The genetics is quite different from what we have in this family, because it's only missense mutations were reported so far. Both biallelic and monoallelic mutations were reported, and the mechanism is supposed to be a loss of function. On the phenotype side, it's also different from Parkinson's disease, because patients had an early onset disease with hyperkinetic movements, and usually in Parkinson's disease it's mostly hypokinetic movements. But what we know from the literature is that one patient who started the disease at 5 years old with chorea had at 60 years old a Parkinsonism. So this is where PD10A and Parkinsonism are related. And in the biology, many studies have explored the link between PD10A and Parkinson's disease, not only 1, but 2, 3, 4, 5, 6, 7, 8, etc. A lot of studies. So we were very interested in this gene, and this is why we performed the segregation analysis. And as you can see here, it was quite disappointing, because with 8 patients who had the heterozygous deletion, only 4 of them had Parkinson's disease. But we didn't give up, and I called back the family to see if maybe we had missed some symptoms. Some patients had Parkinson's disease, but we didn't know. And I don't have time to go too much into the details. But as you can see, almost every patient with the deletion have a phenotype of Parkinson's disease or a neurological disease, such as essential tremor, Lewy body dementia, or schizophrenia. And very interestingly, one of the patients has a widespread dyskinetic choreiform movements in the limbs and the jaw, which reminds the phenotype already described in the literature at this stage. So this story is to be continued. We are still performing the segregation analysis, and we hope, if I come back next year, let's say, maybe I will show you that definitely it's a new PD gene, hopefully. So I would like to wrap up. So we've seen here that long-read sequencing is able to unravel the complexity of genomic rearrangement in already known PD genes such as Parkinson. And second, we also we've seen that with long-read sequencing, we can discover new genes of Parkinson's disease, which is very important for those huge families with no diagnostic up to this day. So with that, I would like to thank the corticoval team at the Paris Brain Institute, which is led by Alexis Brice, and also the NICE CAR team with Cornelis Bauernhardt and Kimberly Billingsley. and thank you for your attention.